Let us now have a quick overview of the basic elements that regulate transcription. A typical, relatively simple eukaryotic gene will have a transcription unit, highlighted in red here, corresponding to the sequence of the primary transcript, from the cap site to the polyadenylation site. This will be divided in exons, in light blue like C, and in introns, the black and white segments in between exons, like D. The exons will make the mature messenger RNA, in reddish brown, which in turn will contain a central coding sequence in green. However, the gene also comprises other DNA regions besides the transcription unit. That means regions that do not become RNA, and that are regulatory, like A and B. Apart from the introns, and the distance between the regulatory sequence and the transcription start site, this schematic is not very different than what happens in bacteria. If you remember, for instance, your lectures about the bacterial operon from last year. However, let us not forget that eukaryotic transcription is far more complex than prokaryotic. Chromosomes are much larger and reside in a confined space, where they need to be packed into chromatin. The level of condensation of chromatin determines whether the DNA can interact with the proteins that regulate the transcription of individual genes. A host of factors are devoted to decide which areas of the chromatin can be accessed, in which cell, and when. But we will talk about the regulation of chromatin condensation in the next lecture. For now, let us focus on two elements. DNA segments that promote transcription, like A and B in the previous slide, and these are called cis factors because they are of the same kind as DNA, which is regulated. Then you also have proteins that bind to these DNA segments and help to initiate transcription. These are called trans factors because they are made of a different type of molecule, and these are generally called transcription factors. So let us start talking about the cis factors, segments of DNA that are required to activate the transcription of genes in certain conditions, like cell type, space, time, or response to stimuli. Here in the picture you can see the expression of the gene even skipped, or EVE. This is expressed in every other body trunk segment in the embryo of Drosophila melanogaster. As you can see in the figure, researchers have managed to identify distinct regions in the genome that are responsible for the expression of specific bands in the body. This is because they contain binding sites for specific combinations of DNA binding proteins that act as transcriptional activators or repressors, which only exist in the appropriate positive combination in those bands of cells. But how can we identify these regions as in the schematic? The gold standard method to identify a region as an enhancer or a cis-regulatory module is through reporter assays. In these assays, you clone a large piece of DNA which you suspect can have enhancer or promoter properties and put it before a reporter gene, which in this case is a thymidine kinase gene. If this segment has the capacity to direct the transcription of thymidine kinase, as it is the case here, you can narrow down the smaller regions responsible for this activity. You do this by systematically substituting small pieces of sequence for a non-related sequence. For instance, you can use the scrambled version of the same number of A, G, Cs, and Ts. Then you scan with this window the initial large piece of DNA for regions that, when you substitute them, diminish the ability of the DNA segment to direct transcription. These regions will be the cis-regulatory elements here in dark brown. In other studies, instead of substituting the segments, this has been done by deleting them or making point mutations when the binding sites were known. But how do you evaluate in the first place whether a reporter gene is being expressed or not? How do you measure transcription? One way is to directly assess the abundance of messenger RNA molecules. You can do that by extracting RNA from the cells that have your reporter, reverse transcribing into cDNA, and then use quantitative PCR methods to have a relative measure of messenger RNA abundance respect to a reference expression level. You can see an additional video that explains qPCR in detail in Moodle. Another way is to use reporter genes that can be visualized by histological methods, such as immunochemistry. This is particularly useful for developmental biology and requires making a transgenic animal for your reporter and then assessing the expression of the reporter gene in the embryo. But if you are using cells in culture, you can also use reporter genes that can produce their own signal, like fluorescent or luminescent proteins, and then measure the amount of light using plate spectrophotometers or a fluorescence microscope. These signals can be quantified with great precision and provide comparative levels of expression. The genomic structure of the gene Pt.2 is a good example to illustrate several of the properties of cis-regulatory factors. Pt.2 codes for a transcription factor that is necessary during development, and a mutation in Pt.2 in humans leads to a condition called aniridia, whereby the iris, the colored part of the eye, is missing. But Pt.2 works in other tissues too. If one looks at the genomic region of Pt.2 in the mouse, there are several regions that are responsible for the expression of Pt.2 in different tissues, like the eye lens primordium, the cornea and retina, but also the pancreas or different areas of the brain. We know this because if you look at transgenic reporters of a DNA segment comprising the two green enhancers, you can see a mouse embryo displaying reporter expression in the pancreas and the lens primordia. If you make another transgene reporting for one of the orange segments, then you can see expression at a later embryonic stage in the developing retina. Having tissue-specific enhancers allows having modular control over the expression of a gene. This is also important in evolution, as these regions could mutate independently and modify the function of a gene by changing the areas in the embryo in which it is expressed, without having to modify the coding sequence or the other areas where the gene is active. Note the scale of the genomic region. Some of these control modules are tens of kilobases away from the promoters that RNA polymerase II will bind to. But if we take a wider view of the genomic region surrounding the Pt.2 transcriptional unit, we can find even more distal control regions. These are shown as colored ovals in the picture. These are now hundreds of kilobases away from the promoters of the gene. Moreover, they are sufficiently far away that other genes, and probably their own control regions too, will be located between these enhancers and the Pt.2 transcriptional start sites. For an enhancer located so far away from the gene to have an influence in the activation of its promoter, that distance needs to be bridged physically. The protein complexes that form around the cis-regulatory region will recruit further proteins or coactivators that will help connect with the promoter, and they will together bring the transcriptional activators with the transcription initiation complex. Some of these protein complexes, like mediator, are dedicated principally to this function, but specific transcription factors can also perform this activity. In different cell types, different enhancers will be active, each making their own bridge. This is a very active area of research, as the enhancer-promoter communication is orchestrated in genome regions that organize in 3 dimensions, and whose organization is not yet well understood. One example of the challenges that enhancers need to solve when communicating with promoters is the sharing of a single enhancer by several genes. For instance, The KIT-SKUT complex in Drosophila melanogaster comprises 4 conserved paralogous genes. They encode VHLH transcription factors involved in formation of sensory organs, like the mechanoreceptor bristles of the fly epidermis. 2 of these genes, KIT and SKUT, have exactly the same expression pattern in the primordium of the fly epidermis, where the precursors of the sensory organs will form. They co-express thanks to individual enhancers that are able to activate both promoters in the same cells. Interesting, similar enhancer sharing can be found in the cluster of globin genes in the human genome.